Bismillahirrahmanirrahim. I will I will explain about C2 oxidative photosynthetic carbon cycle. It is also known as photorespiration. In 1920, the German biochemist Otto Warburg he discovered that whenever there is when the plants they are exposed to the sunlight, they are illuminated. The plants they consume oxygen at higher rate. He also observed that due to this increased rate of carbon dioxide consumption, the photosynthesis was inhibited. Uh, so in normal conditions, the C3 plants, they generally go for Kelvin cycle. But the same plants, whenever they face the hot and dry conditions during the hot summers, what happens that their stomata, they are partially closed during hot and dry days. And then they produce less sugar because the they will not be able to absorb required amount of carbon dioxide from the atmosphere because the stomata will be partially closed, and there will be the declined level of carbon dioxide in the leaves. So that will affect the Kelvin cycle. Uh, in Kelvin cycle, Rubisco helps to add carbon dioxide, but when the hot and dry conditions are prevailing, then Rubisco enzyme, it adds oxygen to the Kelvin cycle instead of carbon dioxide. The product splits, and then instead of 3 carbon compound, as we observed in the Kelvin cycle, the new product will be 2 carbon compound, which is known as phosphoglycolate. And then this phosphoglycolate leaves the chloroplast, and then it moves to the peroxisomes, and then to from the chloroplast to the peroxisomes and mitochondria. So here is the schematic diagram of the normal the Kelvin cycle when the conditions are normal, and photorespiration when the conditions are hot and dry and there is high sunlight. So in normal Kelvin cycle, ATP is used, ribulose biphosphate reacts with carbon dioxide, it fixes carbon dioxide. So the enzyme which is responsible for fixing carbon dioxide is the Rubisco enzyme. And then they will generate 2 molecules of phosphoglycerate. These 2 molecules of phosphoglycerate, then they will generate the uh, uh, glyceraldehyde 3-phosphate, and then sugar molecule will be released. So this sugar consists of 6 fixed carbons. So in case of the Kelvin cycle, 6 carbon atoms, they are released in the form of the sugar, and ATP and NADPH, which is generated, which is produced in the light reaction, and this ATP also, which is produced in the light reaction, that will be utilized here in the Kelvin cycle. So Rubisco enzyme will work uh, as the mediator to fix carbon dioxide, and the reaction is known as carboxylation or carboxylation reaction. But the same plants, when they face hot and dry conditions and there is high sunlight, in that case the Rubisco enzyme, instead of absorbing, instead of using carbon dioxide, it takes oxygen, and then the first product will be phosphoglycolate, which will consist of two carbon atoms, and phosphoglycerate will also be produced. So at this stage, the ATP and NADPH and also ATP from these are produced by light reaction. They will be utilized by the photorespiration reaction itself. So they will not allow the leaves to use this ATP and NADPH for the production of sugar molecule. And at this stage, the end product will be just they will lose carbon dioxide, or they will release carbon dioxide, so they will lose 3 fixed carbon atoms, and the end product will be just that there will be no sugar and no ATP molecules. So here is the general diagram of the Kelvin cycle, normal Kelvin cycle and photorespiration. In normal Kelvin cycle, 6 molecules of carbon dioxide, they are fixed by Robisco enzyme. They react with 6 ribulose biphosphate. So 12 molecules of 3-phosphoglycerate are synthesized. Then they have 12 glyceraldehyde 12 molecules of glyceraldehyde 3-phosphate, and so 2 molecules of glyceraldehyde 3-phosphate, they will produce 1 molecule of glucose, so they will gain 6 fixed carbon atoms. So 1, 2, 3, 4, 5 and 6 carbon atoms. So two Kelvin cycles, they will synthesize 1 glucose molecule, and rest of the 10 molecules of glyceraldehyde 3-phosphate, they again return in the Kelvin in the cycle, and then they generate 6 molecules of ribulose biphosphate. When there is high sunlight and the conditions are dry, in that period, the 6 oxygen molecules, they are taken by Robisco enzyme. The stomata will be partially closed at that period. So these 6 molecules of oxygen, they react with ribulose biphosphate, and they produce 3-phosphoglycerate, which is 3 carbon compound, but only 6 molecules will be generated, while here 12 molecules were generated in the Kelvin cycle. But they will also generate 6 molecules of a new compound, which will be phosphoglycolate, and it has only 2 carbons there. So this is also known as salvage pathway, and 3 molecules of carbon dioxide will be released. So the end result will be that they will lose 3 fixed carbon atoms, and no sugar molecule will be generated, no glucose molecule will be generated in that case. So uh, uh, this cycle will run, and then now we go to the text, that the process is known as photorespiration, because when there is high sunlight, and so in the presence of the sunlight, this process consumes oxygen and releases carbon dioxide. It is similar to the respiration. That's why this process is known as photorespiration. But what is the disadvantage of photorespiration? That they generate no ATP and no sugar molecules. Photorespiration is important, especially under the conditions when there is high light intensity and when there is low intercellular carbon dioxide concentration. What is the benefit of photorespiration for the plants? Photorespiration plays a protective role in plants. It neutralizes the damaging products of uh, uh, the damaging effects of the products of the light reaction. And what are these products? These are ATP, which will be continuously generated, and NADPH. So the plants, they need to have some alternative mechanism to use this excessive ATP and NADPH. So when it occurs, when there is low carbon dioxide concentration, and then the Kelvin cycle progress is limited during that time. So on the other hand, it has some advantages, benefits, that it protects the leaves from the damaging effects of the ATP and NADPH, but it is also considered the wasteful process. Why it is considered as wasteful process? Because it does not allow the plants to use ATP and NADPH, which were generated in the light reaction. So what the photorespiration does, it consumes all this ATP and NADPH by itself, and then there will be uh, no end product of the like in the Kelvin cycle, the end product was the sugar molecule, but here they do not synthesize sugars. So this process is also known as oxygenation because it takes oxygen instead of carbon dioxide, and this is catalyzed by an enzyme which is Robisco enzyme. That's why this process is known as photorespiration. And photosynthesis, as we have seen in the diagram, and photorespiration, they work in completely opposite directions, diametrically opposite direction. So photorespiration will result in loss of carbon dioxide from the cell, while photosynthesis will uh, produce the 6-carbon compound, that is the glucose molecule. This reaction, photorespiration or the C2 oxidative photosynthetic carbon cycle, the reaction, it results in the partial recovery of carbon which is lost during oxidation. So here is the detailed cycle that when there is hot and dry condition, so ribulose biphosphate, it passes through the oxygenation reaction. It takes oxygen instead of carbon dioxide and releases 3-phosphoglycerate compound, and another compound, it will release 2 molecules of phosphoglycolate, and this compound has only 2 carbons, so that's why it is known as C2 cycle also. Now hydrolysis occurs. The water reacts with phosphoglycolate, phosphate, 2 phosphate molecules are released, then they the phosphoglycolate, it produces 2 molecules of glycolate. Now this glycolate will be transported to the from the chloroplast to peroxisomes. Now rest of the reactions, they will be continued in peroxisomes, where 2 glycolate molecules, they are oxidized, they react with oxygen, and Then hydrogen peroxide is released, and two molecules of glyoxylate are synthesized. These two molecules of glyoxylate, they will synthesize two molecules of glycine. Now the glycine will be transported from peroxisomes to mitochondria. So the glycines also uh, through the hydrolysis reaction, they will react with water and NAD plus. So the compound will be synthesized, will be serine, but at this stage carbon dioxide will be released and NADH will be released. So then the serine is transported back to the peroxisomes. Serine will synthesize hydroxypyruvate. Hydroxypyruvate will react with NADH and will release NAD plus, and glycerate will be synthesized. Glycerate will be transported from peroxisomes to chloroplast, and in the presence of ATP, this is the phosphorylation step, addition of phosphate group will occur. So ADP will be released and glycerate will produce 3-phosphoglycerate, and then they will enter in the Calvin cycle, but no sugar molecule will be synthesized. So this is overall the reaction of the C2 cycle or photorespiratory cycle. Thank you very much.